Welcome to the recording on multiple regression. This one will be on the logic and process of what is a multiple regression. As we start to think about regression, we want to remember that it's built on the concept of correlation. And of course, the most important thing about correlation is to remember that correlation does not equal causation. You may have seen this in a previous research class. Maybe even this particular example, that there is a correlation between the number of bars in a city and the number of churches in a city. Could it be that churches cause bars, right? Or could it be that bars cause churches? What? And people will sometimes sit and think about ways in which that might be happening. But in actuality, there's a third variable that's most likely the thing that's causing both bars and churches, and that's the number of people that live in the city. And that those things both correlate with that shared predictor. So we're going to be talking a lot about shared prediction as we talk about regression. So correlation is a basic statistic that you'll need to have a very good understanding and familiarity with in psychology research. So in psychological research, you can get, of course, a range in correlations from -1 to 1, with 0 being perfectly random data and 1 and -1 being perfectly matched data between two continuous variables. In psychological research, we have these general standards. These are taken from Cohen, which is a leader in the regression field. And they would generally say if there is a correlation between -0.25 and 0.25, that's generally considered fairly weak. From 0.25 to 0.40 is moderate. 40 to 60 is probably moderately strong, and anything that's 0.60 or higher is considered quite strong in psychological research particularly, but it's influenced by your area of study. So for instance, I did some health behavior change research in graduate school to help fund my education, and it's very difficult to see change there. Even correlations of 0.25 might get people excited about it. But if you were in a lab and you were trying to boil water with heat, you would hope that the correlation would be higher than 0.60 between heat administered and the boiling of water. So your literature review when you're doing real research will inform you as to what is something people get excited about when looking at levels of association like you see in correlations. A really important thing when you move into regression and correlations is that p-values and significance are no longer the thing that's important. So in my class up until now, we have been doing t-tests and ANOVAs and ANCOVAs and MANOVAs and MANCOVAs, and for all of those, the p-value is very important because you want to look at probability of difference between groups. We are now changing into a whole nother area of statistics for regression. And in regression, it's much more about the strength of association, how strong the relationship is. It is not a big deal about how significant a relationship is. And even conversely, a relationship can be weak but actually significant, and this is influenced by the size of the sample. So if you have a large sample or a very large sample, even the weakest of correlations will come up as less than p.05. And for small samples, it's really hard to get a strong correlation. So you have to pay attention to the significance and not jump to conclusions and not end up making a type 1 error. Right? So for large samples, it's easy to achieve significance, and one must pay attention to the strength of correlation to determine if the relationship explains very much. This is one of the key mistakes that I see students make both in my class and in their dissertations around regression, is that they focus on the p-value of 0.05 and saying that the regression is significant, and this really shows that you're a novice, that you don't really understand the area of regression. That's information that you'll want to look at, but really what's important is the R value, right? The actual strength of association value. So moving into regression, after you get past correlation, you'll want to know that every regression, what's happening is it's trying to find the best fitting straight line, called the regression line, for a set of data. Remember that regression is always continuous data. So what I'm showing you here in this picture is a simple regression, meaning there's one predictor, usually on the x-axis, and there is one over here, outcome, usually on the y-axis, right? So you have your predictor down here and you have your outcome here, okay? And you can plot every person then all along this, field of data. And this is a beautiful bit of data, right? It's a nice hot dog. Everything is together. And so you have all of these points of data. And what statisticians will do is they will go in and they will ask the computer nowadays. In the old days, they did it by hand, which I can't even imagine. But and they would iterate it, meaning you can move this line back and forth until you get the best fitting line all the way down here. Okay, this best fitting line means that you have minimized the distance between the line and all the points of data. And so you have the best fitting line. Now that it's a line in a field, you can actually start to run statistics on it. So I am now going to take you to reach back into the dusty, dustbin of your memory, back into probably 8th grade math for something that you may not have appreciated then, but is going to resurface now like the ghost of 8th grade math. And here it is. The ghost of 8th grade math is the equation for a line. If you remember back in 8th grade, maybe 9th grade math, you learned that y equals mx plus b. And that was probably just drilled into your brain repeatedly. And you learned about slope and y-intercept. And you thought, this is strange information for me to have to learn, but I want to pass 8th grade. So I am going to learn it, and then I'll never have to think about it again. But it's back. All right, so the m and the b are kind of arbitrary variable names, and they're used in the algebra tradition that way. Y equals mx plus b. So x would be the intercept with the x-axis, and y is the intercept with the y-axis, and m is the slope of the line, and b is the where the line intercepts the y-axis. And with that information, you can plot any line in any field of data, right? But the m and the b are variables for algebra, but they're not what statistics uses. So statistics tends to use B1 and B0. Sometimes other variables are thrown in there just to make things very exciting. But in this bivariate regression, you have the same concept, right? So you have the slope, which is paired with the x value, right? It's the regression coefficient, and often called B1. And then the intercept, which used to be y equals mx plus b, is B0, and that would be your y-intercept, right? And then you still have x1, which is the value on the x-axis, and y1 is the value on the y-axis. So it's the same concept that you learned in algebra. So this line that has an equation that represents it, well, if we know that equation, then we can actually take that line and make predictions about what participants should score on an outcome variable if you know the predictor variable. So for instance, if you are looking at neuroticism, predicting the number of therapy visits that somebody engages in, and you collect a set of data and you create a regression line out of it, then we would know, hey, if Jimmy, this new person, comes in and they score a 10 on neuroticism, then they should need however many therapy visits is on the y-axis that corresponds with the 10 on the x-axis, because we know the slope and we know the y-intercept. And in fact, we can even create confidence intervals around that prediction so that you can say with 95% confidence, for instance, that Jimmy should score a 35 to a 45 on the number of therapy visits if he comes in with a 10 on neuroticism. So an important thing to understand, and a caution about regression in general, is around the issues of research design. Regression can often imply to researchers that there was an experiment that happened. However, it's not necessary for there to have been an experiment to have a regression. You could have kind of natural variables as they happen in the wild, and there was a non-experimental design. No one was assigned to any kind of random groups or anything like that, but that you're just looking at how things occur in the natural world. And if that's that how what's happening, then you want to be really careful not to use causal language. So you see this show up a lot more in regression than in ANOVA tradition, because regression tends to be what's used for natural variable analysis and for non-experimental designs. And so you tend to use words like predictor variable and outcome variable instead of independent and dependent variable. And you'll see me start to change my language, and most of the time I'll use predictor and outcome variable, because regression may or may not be an experimental design. In fact, sometimes it's hard to get a regression with an experimental design to it, because people have to be randomly assigned to the predictor variable, which is continuous, and that's hard to do. But your textbook does use independent and dependent variable often, so I just want to point that out so that you're clear and understanding that this isn't something that everybody agrees about, but it really has to do with understanding the design behind the study and making sure you use the correct language to go with the design. Okay, what I have here is a regression line. This one is going in the negative direction, so as x goes up, then y goes down, right? So it's a negative correlation or negative relationship, but this would be perfect prediction. This would happen, you see all the dots are exactly on the line, meaning if we know their score on x, then we definitely know what their score on y would be. It would always be right there, right? And there would be no other option. This would be perfect prediction. Doesn't happen in the real world. In perfect prediction, you can always tell what the score on the outcome variable is for each and every participant if you know their predictor variable. So if we have perfect prediction, then everyone who scored a 10 on neuroticism would all need 40 therapy visits exactly. However, this does not happen in psychological research. We tend to get in psychological research something more like this picture, where you can see the dots, they're clustered around the line, they're not too far off from it, and we can build a nice straight regression equation, a nice straight line, so it's fitting the linear pattern. It's not a curve, so we're happy with that. And what psychologists do, and statisticians and various social sciences in particular do, is they'll build confidence intervals, and you can build them out. You can have them really nice and close with 95% confidence. You can build them further out and do like 90% confidence. But basically saying we are sure that anyone who scores a 10 on neuroticism is going to be between here and here, right? It's going to be in this range from here to here. We're going to catch 95% of people or whatever. And you can get closer and you get less confident that you've captured everybody. So these really nice predictions that people do and the use of confidence intervals within regressions and correlations can really help. And SPSS can actually give you the confidence intervals for correlations. And that's one of the options, and it's a really nice thing to see. So we're looking for fairly accurate prediction. That's really the goal in any regression, is we're going to have a regression analysis to try to tell us, to indicate how close our data follows the regression line. That's one of the major questions we have of the data. How close are all our dots to the line? We look for scatter about the line of all the dots. And the closer to the line, the more predictable one variable is from the other. You can get a nice strong correlation, a nice strong regression, right? An R value, it's called for both of those. They both use R. Correlation uses lowercase r and regression uses uppercase R. But those would tell us how close to the line are all our scattered dots. And we can get a good sense of the regression from just the R or R squared value. There are multiple ways to try to look at prediction accuracy, so I'm going to just kind of walk through that. One is to look at a scatter plot. Obviously, this is the hardest way, but it is descriptive, and you can look at your data in a scatter plot, and you can get a sense of where all the dots are in relationship to the regression line, and SPSS will give you that. But what we tend to end up doing is examining several statistics. One is we look at the R, so the Pearson R, typically the correlation between our predictor and our outcome variable. We look at something called R squared, which is to literally take the R and square it, and that gives us the percentage of the variance that the outcome variable can be predicted from the predictor. There's something called a t-test, which is essentially comparing the data that we got and what it looked like compared to completely random data. So there's a t-test that regression will run, and it will compare your data to random data. So if it's significant, that means your data was significantly different than random data. And you can get a t-test for the R. We also look at something called the B, or the regression coefficient, and then there's a standardized version of that called beta. We can also look at confidence intervals on the R, so the correlation, and on the regression coefficient also. So we're now going to move on from a simple regression where we have one predictor and one outcome variable, and instead talk a little bit about multiple regression. Multiple regression is just like simple regression, but now we can do two or more predictor variables, which is very helpful because most things are not predicted by a single predictor to get your outcome. In the real world, we usually need many variables to predict some kind of outcome. So the question that a multiple regression can answer is how much of the outcome variable can each of the predictor variables predict individually as well as together. So I want to give you some examples, and we'll use these to kind of walk through the logic and understanding what a regression does. So suppose you want to do a study and you want to predict how forgiving people are, and you want to see whether personality factors predict how forgiving somebody is. And you might look at neuroticism and agreeableness because in the past research, those two things have shown up as predictors of forgiveness. So you might want to do both neuroticism and agreeableness in your study and see together how much do they predict forgiving. But you might also want to know how much neuroticism predicts forgiving all on its own and how much agreeable predicts on its own, and which of them predicts it better and to what degree, right? How much weight or degree do each of them have? The strength of association. A multiple regression could look at this. Since we have introduced two independent variables, and they're represented here by the red and white dots in front of you, so two independent variables, these two variables can either not correlate with each other in the real world, or they might actually correlate with each other. And in the real world, most things correlate with each other, any two variables, even if a little bit. And two personality variables, like neuroticism and agreeableness, they're going to correlate with each other some, right? Even with the way in which we ask the questions is going to cause some level of correlation. So you're going to get some overlap. And this picture here gives you a picture of what correlation looks like. There's this overlapping variance for the two variables, and then this is unique variance, right? So this kind of gives you a sense of this is the unique variance for one, and the yellow, and the green for the other, and then this kind of yellow-green in the middle is the overlap or the correlation of the two. And this would be a picture of three variables, and in three predictor variables, there's unique variance, there's shared between two, so each of them share with each other, right? The two of them. And then there's a shared variance in addition with all three of them together. And so you have to be able to understand when we are studying multiple regression, we are studying something that looks like this. We are not studying two separate, completely independent predictor variables. They are not completely independent from each other. They're going to correlate to some degree. In fact, we get concerned if they correlate too highly and it's all the yellow-green in the middle and there's hardly any unique variance, and then we have a problem of multicollinearity that's a threat to our regression. But some correlation is going to happen and we just have to decide how to handle it, and there are multiple ways to handle that. We're going to walk through that. I like to think about the independent variables or the predictor variables in a regression like Play-Doh. So if you ever played Play-Doh as a child, they always start out with like 4 different colors like this, right? 4 unique, beautiful colors. But after you play with it for a while, you start to get some mixtures, right? Where these two you mix together, and these two you mix together, and these two, and these two, and then you have something that had all 4 of them together. It looks kind of brown, and you can't really undo it, right? Like the brown, is it mostly yellow, mostly red, mostly green? How much is contributing to each? Well, I know you could probably get some kind of strange atomic level measurement and separate it out, but let's pretend you can't, because in like our world in the playground, you can't. So you can't undo that. So you want to think about independent variables almost as though you've got some blue and green and red and yellow, but you also have this like red, this pink, or this brown, or you have some different overlapping variances too. And once they've come together, you can't undo it. You have the overlap. So because most of the time variables correlate at least a little, you almost never see a correlation of 0.00 and predictor variables. So then you have to do some work in a regression to examine how much they each uniquely contribute to the outcome and how much them together contributes to the outcome, and pick apart. So if you have 2 predictors, then you have 3 components to analyze: their individual prediction and then their overlapping variance. And if you have 3, you actually have 3, you have a whole bunch of things to analyze. And so we'll talk a little bit more about that. Okay, so I now use a metaphor of sponges and a pail of water, okay, to try to understand how we handle this. I want you to imagine that your 2 predictor variables are like 2 sponges. But in actuality, the sponges came from an original sponge where they have overlap, right? So you have one sponge, right? And let's just say right here you have a sponge, right? That's unique variance. And then over here you have this sponge, and that's unique variance. And then you have this overlapping middle part where the 2 sponges are connected to each other and you can't separate them out from each other, right? That's your overlapping variance. So how do you handle the sponges and the water? Think of the water as the amount of variance and the outcome measure. So this is our outcome measure over here, okay? And the sponges are your predictor variable. You could take all your predictors, all the sponges, their overlap, and the unique variance, and just throw them all into the pail all at one time. That would be one way to handle the predictors. If you decided to throw all of the sponge pieces together and the unique and the shared variance together, this would be called a standard multiple regression, sometimes called a simultaneous approach to multiple regression. In this, all the predictor variables get an equal shot at predicting the outcome variable, just like all the sponges kind of have an equal shot at sopping up the water in the pail all at one time, right? And so this is called a standard multiple regression. It's probably the most common type of regression, multiple regression that's run. But sometimes we might have a theoretical reason why one of the predictors should be given first dibs at the variance and the outcome variable, and the second one goes second. So for instance, say you are introducing a new variable, and maybe we know that neuroticism predicts forgiving, but we don't know anything about agreeableness, like no one studied it yet. And so what you might do is say, okay, I'm going to throw neuroticism in first, and all of it and its shared variance together, right? And I'm going to take it and I'm going to toss that into the bucket first and see how much variance it can predict, right? And then I'm going to take the remaining unique variance all on its own of agreeableness and toss it into what's left of the variance in the bucket. And then we'll get a sense of how well does agreeableness all by itself, with nothing overlapping with neuroticism, how well can it predict forgiving? And that is an interesting theoretical question that can only be answered with a hierarchical regression.